Welcome to the deep dive. Today, we're diving headfirst into a topic that, well, it isn't just at the forefront of biological innovation, it pretty much is the forefront. CRISPR. If you've been hearing whispers about gene editing changing everything, CRISPR is right at the heart of that conversation.
Absolutely.
So our mission today is to pull back the curtain on this, revolutionary technology. We'll look at its unexpected origins and microscopic life to exactly how it works,
Yeah.
the incredible ways it's already transforming medicine and agriculture, and crucially, the really profound ethical questions it's forcing us all to confront.
Right.
By the end of this deep dive, you won't just know about CRISPR, you'll hopefully understand its true power and what it means for our future.
And what's truly remarkable, I think, is the sheer velocity of this scientific revolution. I mean, it wasn't that long ago we barely understood what CRISPR even was.
Right.
And now it's a foundational tool in labs pretty much globally. This rapid progression, it isn't just about speed. It kind of reflects how quickly breakthroughs can empower us to tackle enormous challenges, from human health to environmental sustainability.
Okay, so let's unpack this. It sounds like CRISPR started as, well, a total mystery. What was the biggest head scratcher for scientists in those early days? Was it just random genetic noise they were looking at when they first spotted these odd sequences?
It absolutely was a puzzle. Yeah. Imagine looking at a series of repeated letters in a book, interspersed with totally unique sentences, and having absolutely no idea why they were there.
Okay.
That's basically what happened back in the 1980s when researchers first observed these clustered, regularly interspaced short palindromic repeats, or CRISPR, in bacteria, just weird patterns.
Right.
It wasn't until the early 2000s, and then this crucial discovery around 2007, that the real aha moment hit. These weren't random at all. They were actually part of a pretty sophisticated bacterial immune
It's
system.
in bacteria, an immune system.
Yeah, believe it or not,
Right.
bacteria were storing tiny snippets of DNA from viruses that had previously attacked them, like little genetic memories.
Wow.
They'd integrate these viral mugshots, basically, into their own CRISPR array. Then, if that same virus tried to invade again, boom, special CRISPR-associated, or Cas, proteins, especially one called Cas9, would act like molecular scissors.
Molecular scissors.
And they'd be guided by an RNA molecule, like a tiny homing beacon, to find and just shred the invading viral DNA. Destroy it.
That's brilliant bacteria essentially developing their own genetic memory system. So, okay, how did we then go from a bacterial defense system to a universal gene editing tool for, well, us? That leap must have been monumental.
Oh, it was monumental. Absolutely.
Right.
And that's where Jennifer Doudna and Emmanuel Charpentier enter the story around 2012. They made the truly game-changing discovery.
The Nobel Prize winners, right?
That's them. They demonstrated that this CRISPR-Cas9 system wasn't just limited to cutting up viral DNA. They showed it could be reprogrammed. You could tell it to precisely edit any DNA sequence you wanted.
Any sequence, not just viruses.
Any sequence. And what's particularly striking is how relatively simple and versatile this new tool was compared to what came before it.
Like those zinc fingers?
Exactly. Zinc finger nucleuses, talons. Those were incredibly costly, very labor-intensive. Before CRISPR, gene editing felt like, needing a supercomputer in every lab. CRISPR gave us the equivalent of a laptop. Suddenly, it unleashed this wave of innovation by putting advanced genetic tools into the hands of thousands of researchers, not just a select few.
Democratizing it, essentially.
Precisely. That fundamental simplicity, coupled with its immense power, is why their work, published in Science, earned them the 2020 Nobel Prize in Chemistry. It really changed everything.
So, after all that history, my biggest question is, how does this incredible system actually work at the molecular level? How do we go from that bacterial defense to these molecular scissors that can edit our own genes?
It's actually elegantly simple once you break it down.
Yeah.
You've got the two core components,
Yeah.
the Cas9 enzyme, which is the actual scissors, and a guide, or RNA, or gRNA. Think of the gRNA as the GPS navigator.
Okay, a molecular GPS. I like that.
Yeah. The gRNA is specifically designed by scientists to match a unique target DNA sequence, say a faulty gene you want to fix. It then precisely guides the Cas9 enzyme to that exact spot on the DNA. Once it's there, Cas9 creates a double-strand break. It literally cuts both strands of the DNA molecule.
Okay, it makes a cut. Then what?
Right, now here's where the cell's own natural repair mechanisms kick in. The cell doesn't like having broken DNA.
And the cell has its own repair mechanisms. How do those work with CRISPR? Are there different kinds?
Exactly. Think of it like repairing a broken zipper on a jacket. There are two main ways the cell tries to fix that DNA break.
Right.
One is called non-homologous end joining. or NHEJ. This is kind of like just forcing the two broken sides of the zipper back together quickly, but maybe not perfectly.
A bit messy.
Yeah, it's often messy. It can introduce random little insertions or deletions, mutations, and typically leads to a non-functional repair. It effectively breaks or turns off the gene at that spot.
Ah, so you can use that to disable a gene you don't want.
Exactly. But the other method, homology-directed repair, or HDR, is much more precise. This is like having a perfect replacement piece to seamlessly mend the zipper.
How does that work?
Well, if scientists provide a DNA template, a correct sequence alongside the CRISPR system, the cell can use that template as a guide during repair. It allows for precise correction of a mutation or even insertion of new genetic information.
So using those two cellular repair pathways, this incredibly precise mechanism lets scientists either, 1, disable specific genes by messing them up with NHEJ,
Right.
2, correct existing mutations by fixing them accurately using HDR in a template, or 3, even insert entirely new genetic material into a genome, also using HDR. That really does cover a lot of ground, doesn't it?
It absolutely does. And the toolkit is only getting bigger and better. We now have variants like CRISPR-Cas12, Cas13. These expand the system's capabilities. Some can target RNA instead of DNA. Others can achieve even more precise types of edits, like changing a single DNA letter. And this continuous innovation, it just keeps making gene editing even more accessible and versatile. It keeps lowering the cost and the technical barriers for labs worldwide. It's really democratizing force in genetic research.
Okay, now let's shift to the real-world impact of CRISPR. This is where it gets really interesting for a lot of people. When we talk about transforming medicine, what are some of the most profound applications already happening? Or very close?
Oh, the impact in medicine has been, well, nothing short of monumental already, especially in treating genetic disorders.
Like sickle cell.
Exactly. A huge landmark moment came in 2023 with the FDA approval of Casgevy. That's the very first CRISPR-based therapy officially approved.
The first one.
The first one. It treats sickle cell disease by editing a patient's own hematopoietic stem cells. Those are the cells in your bone marrow that make all your blood cells.
Okay.
They take them out, edit them to correct the mutation causing sickle cell, and then put them back in the patient. The patient can then produce healthy red blood cells. It's potentially curative, truly life-changing.
That's incredible.
And similar approaches are now in clinical trials for other really debilitating genetic disorders, like beta thalassemia, cystic fibrosis, muscular dystrophy.
Yeah.
The list is growing.
That's amazing progress. But for a therapy this groundbreaking, what about accessibility? I mean, is it going to be available to everyone who needs it, or does the cost remain a significant hurdle right now?
That is a crucial question, and you're right, it's certainly a major challenge. While the scientific breakthrough is absolutely immense, the cost of these highly personalized gene therapies is, well, it's very high. Making sure there's equitable access is a huge ongoing discussion for healthcare systems globally. It's something we absolutely have to figure out.
Yeah, absolutely.
And beyond just inherited genetic disorders, think about cancer treatment. CRISPR is revolutionizing how we fight tumors there, too.
How so?
It's being used to engineer T cells, our body's own immune fighter cells, for CAR therapies. CRISPR makes them much better at seeking out and destroying specific cancer cells.
Making immunotherapy even more powerful.
Exactly. For example, trials in 2024 showed real promise using CRISPR to disrupt specific genes that cancer cells use to sort of hide from our immune system. It essentially unmasks them so the immune system can attack.
That's clever.
And then there's diagnostics. CRISPR-based tests are proving incredibly versatile. We saw them develop super fast during the COVID-19 pandemic to rapidly detect the viral RNA.
I remember those.
Yeah, and by 2025, these diagnostics are being adapted for other major global health challenges, things like tuberculosis and Zika virus. They offer the potential for quick, accurate, and potentially affordable detection, even in remote areas.
That's truly astounding, the breadth of it in medicine. Okay, let's move from human health to food security. How is CRISPR transforming agriculture? Is it just about making, I don't know, bigger tomatoes, or is something more fundamental happening there?
Oh, it's much more fundamental than just bigger tomatoes, although better tomatoes are part of it. In agriculture, CRISPR is really revolutionizing how we approach food security and sustainability. By precisely editing crop genomes, scientists can significantly enhance yield, improve disease resistance, boost environmental resilience.
It's like resistance to drought.
Exactly. We've already seen CRISPR used to develop drought-tolerant rice and wheat varieties. That's critical for regions facing increased water scarcity due to climate change.
Huge implications there.
Absolutely. And remember those tomatoes. In 2024, CRISPR-edited tomatoes with higher nutritional content actually reached markets in Japan.
Wow. Already on the market.
Yeah. The technology also lets us create pest-resistant crops, which could dramatically reduce our reliance on chemical pesticides. That's a big win for the environment.
Definitely.
In the U.S., you're seeing gene-edited soybeans with improved oil profiles gaining traction. And there are even CRISPR-edited mushrooms that resist browning, so they have an extended shelf life, which helps cut down on food waste.
Okay, but you hear gene-edited crops, and people immediately think GMOs. What's the distinction here? Is that GMO stigma still hard to shake, even with this precision?
That's a great point and a really important one for public perception. What's key here is the distinction from traditional GMOs, which often involved inserting genes from other species.
Right.
CRISPR edits are incredibly precise. Often they just make small tweaks within the plant's own DNA, changes that could potentially happen through natural mutation anyway, just much, much faster.
I see.
So since they don't necessarily introduce foreign DNA from another organism, many countries, like the US and Japan, tend to classify them differently than old-school GMOs. This often leads to, well, better public acceptance, maybe smoother regulatory approval.
They're not everywhere.
Not everywhere, no. The EU, for instance, has much stricter regulations, often treating them similarly to traditional GMOs, so that difference definitely impacts adoption and trade. And you're right, the GMO stigma can still linger, which is why transparent communication about what the technology actually does is absolutely vital for building consumer trust.
Makes sense. And what about industrial biotechnology? Are there applications there too? Are we talking about like creating entirely new materials or something?
We are talking about optimizing biological processes. In industrial biotech, CRISPR is transforming how we engineer microbes like yeast or bacteria to produce valuable compounds more efficiently.
Like biofuels.
Exactly. For example, CRISPR-edited yeast strains are now being developed and used to synthesize bioethanol, fuel, from agricultural waste products. That directly supports sustainable energy goals by converting waste into something valuable. It's basically about fine-tuning these tiny biological factories.
That's truly diverse. Okay, one more area. How about environmental applications? What can CRISPR do there? Could it help control invasive species, perhaps?
That's one of the most compelling, though also one of the most complex, potential environmental applications. Yes, CRISPR is being actively explored for controlling invasive species.
Like mosquitoes.
Mosquitoes that spread diseases like malaria, Zika, dengue. That's a major target. The idea is to edit their genes to, say, make them infertile, or make them resistant to carrying the pathogen in the first place.
That gene drive, basically.
Potentially using gene drive technology, yes, to spread that trait through the population. The goal is to reduce disease transmission without resorting to widespread, often harmful pesticide use.
Has it worked?
Well, trials in 2024 have shown some success in reducing mosquito populations in controlled, contained settings, which is very promising.
But
Scaling these efforts up to release into the wild raises significant ecological concerns that need really careful consideration. What are the potential unintended consequences? Could it affect the food web? These aren't trivial questions.
right, you're altering an entire ecosystem, potentially.
Exactly. Huge power.
So with such immense power, it seems absolutely inevitable that there would be some equally significant ethical questions and challenges that come along with CRISPR. Where do you even begin with those? It feels vast.
It is vast, and you're right, this really raises the other side of the coin.
Yeah.
CRISPR's power is inescapably coupled with some very complex ethical dilemmas.
Like the situation in China a few years back.
Precisely. The global outrage sparked by that 2018 announcement of CRISPR-edited babies in China, where embryos were reportedly modified to resist HIV, really thrust these concerns into the global spotlight.
Germline editing.
Yes, germline editing. Altering DNA in sperm, eggs, or embryos, which means the changes would be passed down to all future generations. That raises profound questions.
Like what?
Well, the real ethical tightrope here isn't just if we can alter human heredity, because maybe we can, but who gets to decide what counts as an improvement versus a disease? Who decides what's optimal? And maybe more worryingly, how do we prevent a future where access to these genetic enhancements deepens societal divides? Could it create a new form of inequality based literally on our genes? Designer babies, essentially.
It sounds like science fiction playing out in real life. How are different parts of the world responding to these ethical complexities, especially in terms of actual rules and regulations?
Well, there's broad consensus against germline editing right now. Most countries currently ban editing human embryos for reproductive purposes.
Okay.
But beyond that, the regulatory frameworks vary significantly worldwide. What's permissible for research in one nation might be strictly forbidden in another. This patchwork complicates global oversight and collaboration.
And what about in agriculture? You mentioned the GMO debate.
Right. In agriculture, too, there's that ongoing debate about how to classify CRISPR-edited crops. As we discussed, the U.S. and Japan often treat them as non-GMOs if no foreign DNA is introduced, making development easier.
But the EU is stricter.
Yeah. The EU has much stricter regulations, essentially treating them like traditional GMOs, which significantly slows down adoption and creates trade barriers. And of course, underneath all this, public perception remains a major hurdle. Misconceptions about gene editing can fuel resistance, even despite the incredible precision CRISPR offers compared to older methods.
So, beyond the huge ethical questions, are there also practical safety and maybe intellectual property challenges that researchers are still grappling with?
Oh, absolutely. Safety remains a critical hurdle that requires constant vigilance. The big concern is off-target edits, meaning CRISPR accidentally cuts and alters the DNA at unintended locations, not just the target site. This could potentially lead to harmful mutations, like activating cancer genes or disabling important ones. Yeah. Now, newer tools, things called base editing and prime editing, which are like even more precise versions of CRISPR, are constantly improving accuracy and reducing these off-target risks. But rigorous, thorough testing and validation are absolutely essential before any of these therapies can reach widespread clinical use. We have to be incredibly careful.
Can the intellectual property, the patents?
Yes, that's been a whole saga in itself. From a commercial perspective, the intellectual property disputes, particularly over the foundational CRISPR-Cas9 patents between different research institutions. Well, they certainly slowed down commercialization efforts for a while.
Who owns CRISPR, basically?
Kind of, yeah. Though some key resolutions happened around 2024 that have clarified the licensing landscape for many applications, which is definitely a positive step forward. It's still a complex area that companies have to navigate carefully.
Okay, so looking ahead now, the horizon for CRISPR's potential seems truly boundless despite these challenges. What are some of the most exciting, maybe even surprising frontiers being explored right now?
Well, in medicine, we're seeing some really mind-bending advances being explored. Things like partial reprogramming.
Partial reprogramming. What's that?
The idea is using CRISPR, perhaps indirectly, to activate specific genes, often called OSK genes, Yamanaka factors, which are known to help reset the age of cells.
Reset the age. You mean like rejuvenation?
Essentially, yeah. It could potentially rejuvenate cells and tissues, maybe even extend healthy human lifespans. Trials in mice have shown some significant promise, reversing some age-related conditions. Human applications are still early days, but they're actively being explored.
Wow. That's potentially transformative, doesn't even cover it.
Right. And what's also incredibly exciting is the combination of CRISPR with artificial intelligence, with AI.
How does AI fit in?
AI can analyze vast amounts of genetic data to predict the most effective and safest guide RNAs, the optimal editing strategies for specific genes or diseases. It can basically accelerate the design and testing process for new CRISPR therapies at an unprecedented pace. It could massively speed up drug discovery.
Okay, and what about agriculture and the environment looking forward?
In agriculture, you can start to envision crops specifically designed to sequester more carbon dioxide from the atmosphere, actively helping to combat climate change.
Carbon capturing crops.
Exactly. Or crops engineered to thrive in extreme conditions, intense heat, salty soils, which would be crucial for securing food supplies on a rapidly changing planet.
And in synthetic biology, we're really just scratching the surface of designing novel microorganisms for specific tasks, like bioremediation, using engineered microbes to clean up oil spills or plastic pollution. It sounds like we really are just scratching the surface. But scaling these kinds of incredible innovations will undoubtedly require overcoming those significant technical, regulatory, and societal barriers we talked about. How do we ensure this immense power is used wisely and, importantly, equitably?
Exactly right. This is why ongoing public engagement is so absolutely critical. We need transparent communication about CRISPR's immense benefits, but also about its potential risks and the ethical lines we need to draw. That's how you build public trust and inform sound, responsible policy.
And international cooperation.
Oh, essential. International collaboration will be key to try and harmonize regulations, at least to some extent, and maybe more importantly, to ensure equitable access to these powerful technologies, especially making sure that benefits reach low-resource settings so that this isn't just a tool for the wealthy parts of the world. The benefits need to be shared globally and responsibly.
So from its, humble beginnings as just a weird pattern in bacterial DNA, then a defense mechanism, CRISPR has truly ushered in a new era of life sciences. It offers these incredible tools to tackle some of humanity's greatest challenges, curing genetic diseases, securing our food supplies, maybe even addressing climate change. Its precision and versatility have really solidified its role as a cornerstone of modern biotechnology.
The promise of CRISPR is just immense, it really is. But it absolutely demands that delicate balance of pushing innovation forward while maintaining profound responsibility. It's only by openly and honestly addressing these complex ethical, safety, and regulatory questions that society can truly hope to harness its full potential to genuinely improve health, sustainability, and quality of life for generations to come.
So as CRISPR continues to reshape our world at such an incredible pace, maybe something for you, the listener, to think about is, what specific societal or ethical conversation do you think is the most urgent one for us to be having right now? It's an incredibly powerful tool, and it absolutely requires powerful thought from all of us.